Hi, everybody. I'm Misty Peacock. I'm the director of the Salish Sea Research Center at Northwest Indian College, which is just across the bay. Thank you so much for inviting me to come today, Jenny and Cindy especially, and Michael, thank you for helping me out here. And I just wanted to start off by first acknowledging that the work that I do and the place that we are is the traditional and current lands of the Lac-Timish people, which means people of the sea. And the talk I'm going to give to you today is about sea and seafood and that type of theme. Just a little bit about me. My background is in harmful algae toxicology. And so I actually measure biotoxins in harmful algae in shellfish and other marine and freshwater foods. And I use that information so that people and communities can make policy and management decisions about seafood. We want everybody to have access to safe seafood. And so that's a lot of the work that I do. I'm really lucky that I get to be here. I've been here for 8 years. And one of my favorite things to do is go clamming and take my kids clamming. And Bellingham in the summer is a lot of fun to do that. How many people have gone clam digging? There you go. So this work is the Salish Sea's silent crisis, yessotoxins and shellfish die-offs. And it is funded by both NSF and NOAA. And has anybody ever heard of yessotoxin before the emails that got sent out? A couple of people, okay. One of the reasons is that it's a silent crisis is that there's a little bit less known about it than some of the other marine toxins. It is naturally occurring, which means that humans are not putting it into the environment. It actually is produced by algae. It's an algal biotoxin. It has been around for many hundreds of years. It's typically seen in marine waters in late summer and fall. So kind of in August, September, October around here in Washington State. And until recently, it was lumped in together with the diuretic shellfish toxins. Has anybody heard about diuretic shellfish toxins? Can everybody figure out what a diuretic shellfish toxin, the impacts it may cause on humans? Excellent. There are a few species that produce yessotoxin. It is no longer diuretic shellfish toxin because it actually does not have that impact on humans. And so they're separated it out, actually. It's its own separate group of toxins. This is a picture from California. This is the species Linglodinium, and you can actually see this is a true color image. So this was taken from a low-flying aircraft in kind of middle Monterey Bay, California. And this bottom photo is this bloom. The water is kind of a reddish brown, rusty color. And actually kind of right at the top of the circle, oh, I have a pointer, right at the top of the circle, this is a whale swimming through that bloom. And this is where it was coming up, right? So that's how large of a bloom this can be. These are single cell algae, right? So there's a lot of them there. This is in the millions of cells per liter. So algae and phytoplankton, I'm going to use them interchangeably, but it is a very important organism. It produces between 50 and 90% of the world's oxygen. The rest of the oxygen is produced on land, a lot of it from trees. They live in the photic zone, so they actually need sunlight to grow. There's both marine and freshwater species. This is a micrograph of algae. Each one of these is a single cell organism. Here's an algae or a phytoplankton. Here's one, here's one. You can actually purchase this online on Amazon, which I found really interesting to eat. I'm not sure why, but you can. When people talk about harmful algae, I think that sometimes it can be conflated. A lot of times people think, oh my goodness, there's a lot of this harmful algae out there. They're producing biotoxins. It's really dangerous. There's many thousands of species of algae, and only about 60 to 80 of them are considered harmful. So it's really it's less than 2% of the algae that are considered harmful. They can be harmful in a couple of different ways. Some of them can cause fish kills and shellfish and mammal kills. Some of them can be a nuisance, especially harmful to tourism. If you've ever seen kind of ponds that are super green with that cyanobacteria on them, you don't really want to go swimming in them, and I would suggest that you don't. They can have overwhelming effects on the ecosystem, so they can actually grow so fast that they can deplete oxygen and kill organisms in that way. And then the one I'm going to talk about today is toxins. So these are the algae that produce toxins. So yessotoxin comparative toxicity to other toxins in humans is not that toxic. So these three right here, anotoxin A is also known as very fast death factor, microcystin, these are both freshwater cyanotoxins, so produced by algae, naturally occurring. So is saxitoxin, this is also a marine toxin that has paralytic shellfish poisoning syndrome associated with it. These are all very, very toxic. You can also see up there botulism, pufferfish toxin, sarin gas, ricin, arsenic cyanide, right? These are all toxins that we've heard about. They're really toxic to humans. Way down here is yessotoxin in terms of toxicity to humans. And so this talk is not about a toxin that is going to kill humans when you ingest it, but instead, we're going to talk about toxin and how it's toxic to shellfish. It was actually found to have so little impact on humans, they can't actually determine an LD50, so they can't even determine the lethal dose in humans. And in 2013, the European Parliament actually raised the limit that you're allowed to have in shellfish so that you can eat more shellfish if there's yessotoxin there. So if you've been following the news recently, maybe don't, but if you followed the news over Memorial Day, you may have noticed that marine toxins are having a moment. Off the coast of Washington and Oregon, there was a paralytic shellfish poisoning event. At least 20 people were left violently ill. It was over Memorial Day weekend. They actually had to recall commercial shellfish that had been sold. A lot of harvesters had their recreational harvest license. There was subsistence harvesting happening. Low tides were about 1 o'clock to 3 o'clock in the afternoon. Again, it was a holiday weekend. So people were digging for clams, and it actually ended up being 44 people became ill, and multiple people were hospitalized. Nobody died from this event, luckily. So on a scale of bad to really bad, I'm just trying to place you guys in that thinking, not so bad. This is a picture of what those single cells look like in the water during a bloom. And so this is from Terry King at NOAA, and so this is in Puget Sound that this was captured in 2019. But in terms of shellfish, if everything is bad, this is bad too. And so it's linked to shellfish mortality. It may be a tumor promoter in shellfish. These are some pictures, and I'm going to explain them because looking at them from far away, it's kind of like an ink blot. What is that? This is actually in Rocky Bay, Washington, in Puget Sound. This is a beach during a low tide, and these are clams that have surfaced and are either dead or dying on the beach and are gaping. This is what they look like close up. And so they were all open. This is actually the histology of the clams. And so this is the inside of the gills. And these arrows right here, it's a little bit hard to see, but it's damage. And also, this organism, this phytoplankton, is a cyst former. So you could actually see the cysts inside the clam walls. And so this was from the same event. This was in Rocky Bay, Washington in 2019. And then this is in South Africa. This is actually in abalone gills. And so it's the same organism, and it's the same pattern of damage to the gills. And how does this toxin get into the clams or into the shellfish? And so this is a scanning electron micrograph of Protoceration reticulatum, which is that single cell algae. It actually produces the toxin. Somebody always asks, why does it produce the toxin? We don't know yet. Maybe to kill the shellfish. I don't know. But it produces this toxin, and then the shellfish, which are indiscriminate eaters, which means that they're filter feeders, so they eat everything that comes past them, all the phytoplankton, all the algae that comes past them. They have a siphon, they suck it in, it goes into their gills, they digest it. It's actually, the toxin is in the algae, and the shellfish eat the algae, and it's causing these shellfish mortalities. And so I just want to take a second and center where we are. The Salish Sea is really large. A lot of different communities use the resources in the Salish Sea, including the water, including the shellfish. Shellfish is really important to a lot of people. This map is Deborah Reed's map from 2015, and it shows the many of the subsistence harvesters, First Nationers and Native American peoples here, the Coast Salish people, who utilize clams in their everyday food and harvesting. And this map is the same outline. This is all the water drainage, the freshwater drainage that comes into the Salish Sea. You can see it crosses this international border. And there's a lot of communities all up and down here. And so when I was showing that picture before of Rocky Bay, that's right down in here in the fingers of Puget Sound. And that's some of the work that we're going to be looking at. So I work at Northwest Indian College. Northwest Indian College is a tribal college and university. How many people have been to Northwest Indian College? A couple people? Yeah. It's a really beautiful place. It's very close to Western. It's on Lummi Reservation. And as a tribal college, we have the only standalone marine research lab. I want to give a shout out to one of the audience members here, Dr. Marco Hatch, who was the first director of the Salish Sea Research Center. And you can see that we work with our Indigenous students. It's an undergrad-only institution, and we have students and staff who are doing marine research. This is a phytoplankton net, so when I talk about the actual algae that we're collecting, this is how we're collecting it through the water. So this is a net that goes into the water, and it strains out the water, so you have left is the algae. We also run a summer internship program, and so when we're doing our summer harvests, our students are actually out there collecting the shellfish that is used in this study. One of the funding awards that we have is through NSF, and so that's the T Center for Community Marine Research. And so the work that we do is really focused on community research projects, so identified by the community, supporting tribal nations' food and sovereignty, and having community knowledge exchanges. One of the reasons that we have this award, well, one of the benefits of having this award is that we actually have the instrumentation and the equipment to do these kind of biotoxins in the lab. So everything that I show you happened at the research lab at Northwest Indian College. We didn't send out any of our samples. We did all of the research ourselves as a research group. So back to this map of the Salish Sea. Here is a micrograph of the organism. This is what it looks like when you look at it through a microscope. This is Protoceratium reticulatum, and this is a yessotoxin producer. This one will also color that water, that red color. And you can see it kind of has a red golden color right there, and that's why the water looks red to us. In the Salish Sea, there's been repeated mortality shellfish events in the summer, and it happens to be around the same time that this organism is identified in those net toes. Terry King right here works at NOAA, and she's a shellfish biologist, and Dr. Vera Trainer, who works at the University of Washington, is a phytoplankton ecologist here in Washington State. And they began noticing in 2016 and 2017 that there were these repeated summer die-offs of shellfish at commercial shellfish growers. So commercial growers reached out to them and said, our shellfish are dying. These die-offs have been happening for hundreds of years. There's a lot of local and traditional knowledge about these summer die-offs, and they were kind of assumed to be caused by spawning stress and bacteria. In the summer months in Washington State and in the Salish Sea, a lot of the really low tides happen around noon, 1 o'clock in the afternoon. And so you can imagine if the water goes away, it can get really hot and warm, and also bacteria can start to grow, which is why a lot of times in the summer months for recreational shellfish harvesting, they'll say make sure you cook your shellfish to kill off any bacteria that is there. This is also when the shellfish are spawning. And so it was kind of thought that maybe it was the spawning stress in bacteria where they would have these die-offs. I showed you a picture in that first slide of that one where the whole beach was covered with those shellfish that were dead or dying. And that was kind of an extreme event. That was in 2019, but there have been milder ones throughout the years. But so Terry and Vera thought maybe there's a link between high concentration of the organism, of the algae Protoceratium reticulatum, the toxin, yessotoxins, and summer die-offs. And so they began to investigate this. And a paper was produced from this, Hiding in Plain Sight, Shellfish Killing Phytoplankton in Washington State. And I'm on it, and one of my students is on it as well. Terry King and Vera and other scientists in Washington State and the NOAA Research Lab in South Carolina as well. So in 2021, when this paper came out, it's not silent anymore, right? So a lot of times think of ourselves as the Lorax for the shellfish. We speak for them because they're very quiet. And so I just want to point you to this table from the paper. You can see this is from 2016 to 2017, and there's a few in 2019. And all of these with numbers here are shellfish that had yessotoxin in them that corresponded with these summer shellfish mortality events. And so yessotoxin is probably playing a role in these repeated die-offs, and it is not just spawning stress and bacteria. And so this was in South Puget Sound, impacting the commercial shellfish. You can see most of the organisms that were collected were mussels, oysters, and a few manila clams as well. So Dr. Trainer actually in 2013 wrote this paper, Diuretic Shellfish Toxins. Remember, yessotoxin used to be considered one of the diuretic shellfish toxins, and Jerry Borchert at Department of Health. And so Washington State actually monitors for these species of algae in the water, and they also monitor for these toxins to make sure that humans are safe from these toxins. We now know that yessotoxin is probably not impacting humans very much. But also I wanted to point out, this is a paper from Dr. Charteau in 2023, and he's a Canadian scientist with Department of Fisheries on the west coast of British Columbia, and they also are finding these yessotoxins in the water. So after this paper in 2021, we got together and we thought, what can we find out? What can we learn more about these yessotoxins, these summer mortality events, and everything that is happening in the Salish Sea? And so we began in 2021 to collect summer. We were going to collect from 6 sites. We were going to collect 5 shellfish at each site, and we were going to do it in June, July, and August, just in 2021. But we didn't, because you can see from here that there are quite a few sites that we collected at, and it's through 2024, we are still collecting shellfish. It's 2025 now, but it is cold outside. So this 2021 paper is from down here. This is Rocky Bay. This is where the shellfish were collected in the die-offs that happened in 2016, 17, and 2019. Here's our summer interns and visiting graduate students collecting shellfish up at Birch Bay. And so we split it up, and we had people who were working at the northern sites and people who were working at the southern sites. And all the shellfish were either coming straight back to the lab with us, or they were being mailed to us. We only lost 1 batch, and I feel really sorry for that mailman. But some of the project questions we wondered about were, can we measure yessotoxin in various types of shellfish? And so from that 2021 paper, you saw that most of them were mussels, oysters, and a few manila clams, but we wanted to see if it made a difference which type of shellfish was there. And so then that brought the second question, is there actually yessotoxin in different types of shellfish, or is it just a few shellfish that hold this yessotoxin? Do we need to monitor for yessotoxin separately now that it's not one of the diuretic shellfish toxins? And it is actually consistently causing the shellfish die-offs in the Salish Sea. This is actual footage of one of the techs in the lab, Megan Schultz, who is making clam milkshakes. And that is exactly what they are. This is a clam. This is a sonicator, so it's using sound to break up the cells. We make these beautiful milkshakes. They smell delicious. You want to make sure that you don't have a smoothie for breakfast or lunch. But other than that, that is what we do. And this is how we get the toxin out of the clams. We're actually running on liquid chromatography tandem mass spectrometry. Western Washington University has one of these as well. So if you guys are interested, you can make your own biotoxin lab. But what this does is that actually we are looking for the mass of yessotoxin, and that's how we're running these samples. So we turn them into milkshakes, we extract them, we take the toxin, we run it through some chemistry, column work here. It goes into this big instrument, and we can actually find the mass of yessotoxin and how much yessotoxin there is in each sample. So that first project question, can we measure yessotoxins in many species of shellfish? The first shellfish that we tried to run in 2021, the answer was no. Apparently, shellfish innards are really gross. Not to eat because they're delicious and I love them, but the instrument did not think that was appropriate. And it made our instrument so dirty that we actually couldn't measure anything. We had to call a service tech. And so we took these samples back and we did a whole bunch of cleanup steps before we ran them through the instrument. And then we would spike the samples with standard amount of yessotoxin. So this is a known amount of yessotoxin. And we found for each of these specific type of clam or shellfish that there was a really good recovery. So considered really good recovery is anywhere between about 80 and 110%, and that's just because of instrument error. And so you can see that these all fall within that. And some of them are really great, the ones that are close to 100% especially. Our second question is there yessotoxin in many species of shellfish? These are the species of shellfish in the Salish Sea that we have found yessotoxin in. We have measured yessotoxin in. Most people are probably familiar with butter clams and oysters and cockles. Have people eaten cockles? They're really good. They're very sweet. Mussels, Makoma clam, oyster, varnish clam, softshell clam, oyster, another virginica oyster, native little nut clam, Pacific oyster, manila clams, horse clams, and gooey duck as well. And all of these clams at some point have measurable toxin. So that brings us to the next question. Do we need to monitor for yessotoxin? And this is a tricky question because when we say monitor, are we saying do we need to monitor for the shellfish or do we need to monitor for humans? So I'm going to break that down a little bit. On the bottom of this graph, we have our species names, and some of these are just not all of the species that we found yessotoxin in, but we have oysters and mussels and butter clams, manila clams, cockles, gooey ducks, and horse clams. Right here, we have a log graph. This is yessotoxin equivalents in nanograms per gram of shellfish tissue. When it says below the limit of quantification, as toxicologists, we never want to admit that we can't measure something. And so we say, we don't really trust that that's the quantifiable number because it's so low, but just give us time and we'll make a better method. Down here are true zeros. So these clams have no yessotoxin in them. You can see the colors are based on the species. And you can see there are patterns here. We have our mussels had no zero. Our butter clams, we had quite a few zeros, but we also had some that had low yessotoxin, cockles, gooey duck, and of course, horse clam up here. I want to point out that this is from 2021 through 2024, all of these shellfish, and that they were not all collected on the same date, the same time. I'm going to get more into the timing of that in the next couple of graphs. This is just based on the species that we tested at all of the sites that we had. This right here is the US regulatory limit, 1,000 nanograms per gram of tissue. So for a shellfishery to be closed down in the US so that humans don't eat this shellfish, commercial shellfishery or recreational shellfishery, you would need to have reached this limit or be approaching this limit. And you can see that all of our clams are below that limit. Europe, after 2013, actually made it higher, right? So theirs is 3,750 nanograms per gram, nanograms of toxin per gram of shellfish. So they're way up here because they found that it actually wasn't that dangerous to humans. So in terms of monitoring for human safety, there's a little bit of concern. These ones are approaching that limit, right? So it's maybe not a bad thing to monitor, but you can see that the bulk of the clams that we tested over these 3 years didn't come close, didn't approach that regulatory limit. It gets more complicated because nobody knows what the limit is for killing clams. There's no regulatory limit for killing clams. There's actually no LD50, no lethal dose for 50% of the clams. We have no idea what the concentration is that actually starts to kill clams or when they have those shellfish die-offs. And this is the same type of graph, but now it looks very complicated, but now we have the months that were sampled. This is all the sites again. So that picture of the Salish Sea where we had all those red dots, we had kind of a cluster in the south, cluster in the north, and also San Juan Island, mostly because the interns wanted to go to San Juan Island, which is beautiful. But so we have June, July, and August when we were supposed to test for clams, and then September, October, November, December, and January, which we tested for clams just for fun. And you can see again, there are quite a few zeros. Right here I have the numbers. So these numbers right here are the numbers of zeros. So there's 13 zeros in June, 10 in July, 16 in August, 6 in September, 4 in October, and these are individual clams. And these are the ones that are really low limit down here. But you can see that there is clams in July and August and September. And typically we see the algae in the water in July, August, September, and sometimes into October. So this is probably when you would anticipate seeing the yessotoxin in the clams. And just visually, you can see it increases as you get towards the warmer months and decreases again as you get towards the winter months. But surprisingly, is still present. And this is either because it's in the water, the organism is in the water, or because the clams are holding on to the yessotoxin inside their bodies. So some marine toxins can actually stay inside of clams for multiple years. Here's that map again with all of our sites. And you can see here, we just kind of split it into north and south without rhyme or reason, except for we collected in the north because this is about how much you can collect in a day, and the south, and this is where a lot of the commercial shellfish harvests in Washington State are, and they were sending us their clams. There's 89 samples in the northern section, and there's 53 samples in the southern section. And you can see that there's a variety of it. There's not really necessarily a pattern on southern and northern. There's yessotoxin throughout the Salish Sea. And again, here are these cluster of those 5 sampled, the geoducks, the horse clams, and one of the mussels that had that high toxin. They were all from the South Salish Sea. So those are the questions we had about monitoring. And then we thought, is it actually causing consistent die-offs? So we did, in that 2021 paper, we did show that it was correlated with those die-offs that happened in 2016, 17, and 2019. And we just happened upon a die-off that happened in 2024. So in August 2024, August 5th and August 16th, we were contacted by Claire Scheelman at Lummi Natural Resources, and she's the shellfish biologist there, and also Emma Sauce at Port Gamble Squallum Tribe Natural Resources, and they said that we have clams that are dying. Can you test them for yessotoxin? And we said, absolutely. We went out and harvested the clams ourselves at Lummi Sea Pond, and these clams were on the surface like that first picture. They were dead and gaping on the surface. They had opened up, and we collected them at the Lummi Sea Pond, and they were sent to us from Indian Island, and it was the same there. These clams and other species were on the surface of the water, and they were dead and dying. Lummi Sea Pond, no yessotoxin. Indian Island, yessotoxin. So it probably contributed to this die-off. It did not contribute to this die-off. So our project questions, again, we had, can we measure yessotoxin in various types of shellfish? I'm going to give our lab group a gold star on that. We can. Is yessotoxin present in multiple types of shellfish? We probably don't deserve a gold star for that, because that's not a good thing, but we get one. And do we collectively need to monitor for yessotoxin? Again, that's a little complicated. I would say yes in terms of shellfish, because shellfish is such an important resource for so many different communities in the area, not only commercially, but subsistence and recreationally as well. For humans, probably not. Is yessotoxin consistently causing shellfish die-offs in the Salish Sea? Again, I don't know if we can say consistently. I think that it's contributing to die-offs clearly, but it's maybe not the answer to all those summer mortality events. I want to talk about a couple other ways we're looking at these questions. This is Megan Schultz, and she is a research technician at Northwest Indiana College. She's also a grad student at University of Washington. And she's actually working on another NOAA project we have on climate algae acidification and climate change in the Salish Sea. Harmful algae, not climate algae, sorry. And so she's actually growing the organism. This organism was collected by Rosa Hunter off of Blaine, Washington. And we've been growing it in our lab since 2023. And so Megan is actually determining their growth rates. I'm not going to get too much into the weeds on this, but this is a growth curve of different temperatures. And so she actually is growing them in different temperatures in 10 degrees, 16 degrees, and 22 degrees. The water off of the coast of Washington, Celsius, is typically around 12 to 15 degrees Celsius. And she's actually measuring their growth rates. And you can see that at 22 degrees Celsius, the rate of growth is actually faster of this species. So this is a thought experiment on what's going to happen as the waters continue to warm off the coast of Washington State and the Salish Sea. Again, I'm not going to get too much in the weeds with this, but this is the total yessotoxin that's being produced in those samples that she's growing. And she's taking it at 2 growth points. She's taking it when it's growing strongly. This is called exponential growth. So this is when you have your cells growing really, really fast. And then this is stationary phase, when the cells are starting to die and they stop growing because they became limited by nutrients. And you can see the takeaway from here is this is 10 degrees, this is 16 degrees, and again, this is 22 degrees. That really, when those cells start to die, you have a lot of dissolved toxin and you have a lot more toxin in the warmer grown algae. So this is work for her master's thesis that she's doing. Another project we're working on is Dr. Noir Ayachi. She's at the Virginia Institute of Marine Science, and she came here last summer and spent a month in our lab. And she worked with Jamestown Squalum Tribe, Taylor Shellfish, and Lummi Nation, and actually received a Pacific oyster spat. And those are the little babies. And she was growing them, and I'm going to zoom in on this poster. This work was presented at the United States Harmful Algae Conference, Harmful Algal Bloom Conference in last November. But she wanted to see what would happen to their growth and their mortality when they're exposed to yessotoxin. So kind of starting to get at that question of what is the limit that is needed to kill the shellfish? And so this is zoomed in from this part of her poster. So this is the little shellfish embryos. This is the baby larvae. And you can see that this is the concentrations, nanogram per milliliter. So this is well below what you would consider that regulatory limit that I talked about earlier. But you have mortality increasing as you add more of the toxin. But what's really interesting is the recovery, and that once these young larvae, these young oysters, were exposed to the yessotoxin, they didn't recover. They didn't get rid of it once you removed them from it and continue to grow. They actually died. And so she actually did counts 3 days after they had been exposed, and a large percentage of them had died. She's going to come back this summer and do these experiments again. And so just a few takeaways. This is a lot of years, but in the North Salish Sea, these are all the years that we found yessotoxin in shellfish, measurable yessotoxin in shellfish. Yessotoxins may be a contributory factor. This can also be exacerbated by other things like temperature or nutrient limitation or ocean acidification. And it's not just a commercial concern, right? We're not just worried about people who are selling shellfish, but also people who rely on shellfish for recreational and subsistence harvesting. And then I always find this really interesting. One of the oysters we received has this invasive Japanese oyster drill. And this is a predatory snail, which you wouldn't think so, but it's a predatory snail, and it actually drills a hole through the oyster, injects its mouth parts into the oyster, proboscis into the oyster. I'm a toxicologist, I'm not a biologist. And it sucks out the oyster meat inside. And we happened to have an oyster sent to us that had one of these Japanese oyster drills attached to it, and we removed it and found that the Japanese oyster drill by itself had measurable yessotoxin in its body. So this yessotoxin is moving up the food chain as we go along in the marine environment. And I just wanted to say Haishka, or thank you, to everybody who worked on this in the lab. And then we're going to see if this works. Kara Walters, who is here, she's a WWU alumni. She works in the lab as a technician, did the phytoplankton counts from the net tow today, about an hour ago. And I want you to keep your eye right up here. That is a singular Proceratium reticulatum swimming around. She found one in the net tow because it knew we were giving this talk. So I can play it one more time because it's kind of fun. These actually move around. They have flagella that helps them move around. Swimming around. And that's Kira with her iPhone held up to the microscope's eyepiece. And it is February. Not really the time of month you would anticipate seeing this organism in the water. And that's all I have. So we're trying to figure that out. So as the research I showed where Megan was doing these preliminary experiments, she's actually looking at that. Her next step is to grow them in kind of larger quantities. And so she's growing them at 22 degrees. We're actually going to project them forward. We're going to take the 16 degrees, 22 degrees, and even go probably a step warmer and see how much toxin is produced. We do know that these types of phytoplankton, these algae specifically, grow faster when the water is warmer. So we're not really certain if there's a link to how much toxin they produce, but warming water can stress the algae, and a lot of times algae produce more toxin when they're stressed. I'm going to repeat for Zoom. So the question was, what do you do if you are finding that the yessotoxin causes the die-offs? So we work really closely with our partners. One of the things that we do is we really protect our community's data sovereignty. And so when I talked about the samples from Lummi Natural Resources and Port Gamble Squalum, I had that with permission to discuss that. And so I provide them with that samples. We don't do management or policy decisions, but we provide the information so that each group can do that policy decision. We also work with the Washington Department of Health. And so we're part of a group called Sound Toxins, which is a community monitoring program where we actually go out and take those net tows. And so that data is shared with the state so that they know when the organism is in the water. So Kira will have uploaded that she found 1 protostration in our net tow today. And so then people do that. And if we're given, for example, like commercial growers provide us with samples, then we would partner with them to provide them with the information. Again, the silver lining in this, if it can be said, is that this particular toxin does not seem to impact humans in the way that a lot of other marine biotoxins do. Of course, the concern is to shellfish, but there's less of a human health concern. Marco?
It seems like the impact on adult survivorship or mortality is a bit more mixed, but the larval data looked really bad, right?
Yes.
So that's an interesting one to think about, where we tend to focus on what we can observe, but it seems like a lot of the story is things that we're not seeing, right? Like measuring the larval impact.
Yeah, so Marco's question was that it seems to be mixed on the adult clams, the die-offs, but the larval data looked really bad. And so maybe what's happening is behind the scenes. It's something that we haven't looked for. And I agree with you 100%. I think that this is one of the only marine toxins that really impacts shellfish directly. A lot of times the toxin is passed through and impacts up the food web chain into humans, into marine mammals, into marine birds, and other larger species. And so I think that maybe less study has been done specifically on the shellfish.
I guess I'm curious, are we talking about one structure, or is this a class of structurally related compounds? And I saw yessotoxin equivalence on one slide. Maybe that's related. Could you explain what that means?
Yeah, so the question is yessotoxin just one structure, or is it a class of compounds? Because I talked about yessotoxin equivalency on one of the slides. And so right now there are 4 yessotoxins. So there's yessotoxin, homoyessotoxin, yessotoxin OH45 and homoyessotoxin OH45, which are all in the same class of compounds. They just have different side chains, essentially is the easiest way to explain it. And right now, because there's so little data on lethal dose in humans, they actually consider yessotoxin, homoyessotoxin, which are the 2 that we measure for in the lab. So we're giving total yessotoxin there. And 45 yessotoxin, the same equivalency, they're all equal to 1. And homo 45 yessotoxin has an equivalency of 0.5, because even with the mouse studies, they found that there was no impact on the mice at all. So, yeah. So the question was, are we able to monitor for multiple toxins at once? Yes, absolutely. The method that we're doing here, we look for yessotoxins, and we also look for the other diuretic shellfish toxins. What we are doing behind the scenes as well is that we are measuring for paralytic shellfish toxins, domoic acid, and a suite of 7 cyanotoxins, all from the same samples. But that's a different story.
Dr. Peacock, we have a few online, if you don't mind.
Sure.
Okay, so this one is two questions. Is the yessotoxin in the algae or is it released when digested? Is there a yessotoxin in the water associated with the algae?
Yes. So the yessotoxin, actually this slide that's up right now kind of shows both of that. So the purple is the yessotoxin that's inside the algae. That's particulate yessotoxin. And that's what the clams are eating. So they're eating the algae that has that yessotoxin. The green bars right here on this slide is the yessotoxin that's dissolved in the water. So that's yessotoxin that's outside the body of the algae. There is still some understanding that needs to happen around whether shellfish take up the dissolved portion of toxins as well. The data is mixed right now. Probably they're getting most of the toxin from ingesting the actual algae, and then it's going inside the body. But what can actually happen in the body of the shellfish is that toxin can become some of the other toxins when the question was, is this a group of toxins? So some of those toxins happen inside the body of the shellfish. And we do measure for it in the water's dissolved samples as well. So we have environmental data as well, not just shellfish data.
Well, it's about 20 after. Sure we can stick around down here if people want to come down and have some more questions, but thank you.
Thank you.